you this is one of the most boring topics of pathology. It's extremely dry and it can get irritating at times. Even while teaching this topic, one really feels at wit's end. But we have to read this topic for a very specific reason. So, first of all, tell you why we're supposed to do all this. So the reason why we're interested is that inflammation is actually a very, very important process. As far as your learning objectives are concerned, as per the new curriculum, of course, in the exam, you're going to be asked something like this: enumerate and describe the mediators of acute inflammation. So that's a very straight one-line question, but obviously the answer isn't as simple or one line as what we can see. Now, if we talk about inflammation, the word inflammation was actually derived from the Latin word inflame, which means to set on fire. And inflammation is definitely one fiery business. So again, inflammation is sometimes good, it's sometimes bad, and it's always a little ugly. Now, why? Because inflammation basically results in well, it can cause pain, it can cause swelling. If you have a boil or a pimple on your face, that is also just a form of local inflammation. If you have any allergy, that is again inflammation. And autoimmunity is when the inflammation is completely misdirected so that the body's mechanisms start destroying cells. And of course, because of chronic inflammation, what happens is that you become more susceptible to infections. Your body just kind of weakens down, and of course, you're susceptible to poor wound healing. Now, yes, so there was a cover editorial on Time also that inflammation is actually a secret killer, and secret killer it is because it constitutes a very, very important chunk of human disease. Now, when we classify inflammation, the site of inflammation, as per the site, as I said, acne or a pimple on your face, that's something that's local inflammation. But when the inflammation involves the entire body, it becomes very widespread, and in medical terminology that is called a systemic when it affects all organ systems. Now, if you talk about onset and name of eicosanoids, and they're synthesized by the COXes, that is the cyclooxygenases, which lead to the production of the prostaglandin family, and the LOXes, or the lipoxygenases, which cause the production of leukotrienes and lipoxins. Now, these guys can mediate virtually every step of inflammation. Now, prostaglandins are very, very important. Uh, Basically, they are produced again by mast cells, macrophages, all the extracellular guys, mast cells, macrophages, etc. And prostaglandins are produced by the action of two COXes, constitutively expressed COX-1. Now, what do you mean by a constitutive expression of a gene? A constitutively expressed gene is basically one that is continuously being expressed. An inducible expression refers to an expression that happens in response to a particular stimulus. So these are two types of gene expression, constitutive and inducible. Now, if we look at this, prostaglandins are then, yes, so prostaglandins further have a long list, A, B, C, whatever, but the most important ones with regards to inflammation are DEE, FGH, and I. And then they have numbers in the subscripts, which basically refer to how many double bonds they have in their structure. So the most important ones in inflammation are D, E, F, and I, and there's also called something called thromboxane, which you've studied about. So these are the important ones. And prostacyclin, basically, that's PGI2. It is a vasodilator and it's an inhibitor of platelet aggregation. Now, about one line about the prostaglandins. I'll tell you just the very, very important features which you need to know. Although when you are sitting for an exam, then the question is going to be enumerate and describe. So very frankly, jitna diya hua isme is more than sufficient, because it's slightly difficult to retain anything more than this. Anyway, PGD2 is a major prostaglandin which is coming out of mast cells. Now, PGD2 causes vasodilation and it also causes edema. F2 alpha is easy to remember. F2 alpha is the prostaglandin that is associated with uterine smooth muscle, bronchial smooth muscle. So it's the one that is going to cause uterine contractions. PGD2 is a chemoattractant for neutrophils. Okay, so this is an MCQ. Chemoattractant for neutrophils is, and they'll give you these various options. So that is PGD2. PGE2 is a hyperalgesic and it makes the skin hypersensitive. So it is involved basically in the pain response. Secondly, PGE2 is also involved in cytokine-induced fever during infections. Now, needless to say, this is a totally different topic, but since this has come up, it just came to my mind, the cytokine storm of COVID. Now, that is also important to understand in this context, because if you understand the cytokine storm in COVID is nothing but an inflammatory response, an exaggerated inflammatory response, which was brought about by a set of molecules called as cytokines, especially TNF, and sometimes an exaggerated inflammatory response can lead to a lot of havoc, as we've seen in COVID-19 cases. Now, this is just a small chart that is summarizing the various prostaglandins, the important ones. So PGI2 I always remember as I. So I is inhibiting platelet aggregation and it has vasodilatory effects. F2 alpha, as I said, the uterus. So uterus, uterine contractions. And uterine contractions means that it has the potential to induce labor, and if exaggerated, it can even cause abortion. PGD2, basically, it again is associated with inflammation and neutrophil recruitment, and TXA2 or thromboxane results in aggregation of the thrombocytes. So, coming down to the other branch of the story, and that is leukotrienes. Leukotrienes, as the name suggests, leuko. So it is secreted mainly by the leukocytes. It is produced by the lipoxygenase enzymes, and it serves as a chemoattractant for leukocytes. So there are three different lipoxygenases: 5-lipox, which is the main one in neutrophils, and 5-lipox is the ultimate chemotactic agent for neutrophils. Chemotaxis means that it's basically recruiting neutrophils, it's helping in their movement. LTB4 again is a potent chemotactic agent of neutrophils, and in addition, LTB4 results in generation of reactive oxygen species and the release of lysosomal enzymes. And then you have LTC4, D4, and E4, which are resulting in intense vasoconstriction and bronchospasm. So if you look at all this, we'll have to kind of, intermittently summarize what we're speaking on this topic, because it just kind of evaporates every 10 seconds. So you have cell membrane phospholipids. Now, as I said, arachidonic acid doesn't come free for all. It's bound to the membranes with the phospholipids. Now, how do you release arachidonic acid from the membrane? You have enzymes called phospholipases, and arachidonic acid is then acted on in two enzymes, COX and LOX. COX can be COX-1, which is constitutive, COX-2, which is inducible. And after that, the COX brigade is going to lead to the synthesis of all the prostaglandins. The various prostaglandins, I've told you already, DEF, GHI are the important ones in inflammation. And you have F2 alpha also, which is in the uterus. Okay, prostacyclin is also called as PGI2, and thromboxane has vasoconstrictive effects. Okay, so there are just a few mediators that I talked about till now that have vasoconstrictive effects. One is thromboxane and one is the leukotrienes, CDE. Okay, the rest all are vasodilatory. Then you have the second branch, that is the LOX. LOX activity leads to the production of the leukotrienes. B4 is a neutrophilic chemoattractant. C, D, E, they are basically very important in respiratory pathology. And then after that, there is a sister of 5-LOX called as 12-LOX, 12-lipoxygenase. And this results in the synthesis of the lipoxygens. So this was just a short summary of what was happening till now. I hope this is very clear to you now. So again, I'll recapitulate for you. Chemical mediators, two types: cell-derived, plasma protein-derived. Cell-derived can be preformed. Preformed are the first line mediators: histamine, serotonin. The rest of the pack is all synthesized on the go, and after that you have arachidonic acid metabolites and eicosanoids, prostaglandins, LTs, and now we'll talk about lipoxins. So lipoxins are generated from arachidonic acid by the lipox pathways. So unlike LTs, which promote inflammation, lipoxins are actually inhibitors of inflammation. Now I'll talk about that later. Now, so this is an inhibitor of inflammation, that is, iska kaam hai, that is, it's going to stop the inflammation. Now, so basically leukocytes produce intermediates in all this lipoxin pathway, and this is converted to lipoxins by the platelets. So as you can see, platelets have a very, very important role. Now, what are the principal actions of the lipoxins? Lipoxins, they're going to inhibit inflammation. How are they going to do it? They're not going to let the WBCs recruit. They're going to stop the soldiers from coming to mount the defense response. Secondly, they are going to inhibit chemotaxis. Okay, so first of all, you don't bharti mat karo in logon ko, and then secondly, you don't let their movement happen either, right? Then the next thing is, yes, as I said, LTs promote inflammation, lipoxins don't promote inflammation. So there's an inverse relationship, and hence it is believed that lipoxins play a role in the resolution of inflammation. So Why are we, as I said at the beginning of the session, why are we interested in studying such a dry topic with such long lists of all kinds of mediators that we really don't memorize, you know? The reason is because when you become, or if you are inclined to take up pharmacology later on as a profession, or you land up in research and all these things, and you land up in drug design, it is extremely important for you to understand the nature of these molecules, because all inflammation can theoretically be blocked by designing drugs that are going to work against these molecules. So anti-inflammatory drugs can work by inhibiting the synthesis of eicosanoids. So now let's look at this, COX inhibitors. So if I block the COX pathway by certain drugs called COX inhibitors like aspirin, that's a very common drug, or NSAIDs, painkillers, they're going to inhibit both COX-1 and COX-2. And if we go back to that slide, now if you block COX, you don't get prostaglandins. So that's a very, very nice way of stopping inflammation. The second thing is if you block LOX. Now if you block LOX, you're not going to get the leukotrienes. So you don't have leukotriene production. There's a drug called Xilutron that is used in asthma, and it is based on this philosophy only. The second one is don't block leukotriene production, but block the receptor on which leukotriene is going to come and activate things. So that drug is called as montelukast. Okay, so that is an LT receptor block. So these guys, these drugs are all useful in the treatment of asthma. Then you have broad spectrum inhibitors. If you take steroids, now steroids are capable of blocking every level of inflammation. So you have, they can block COX, they can block phospholipase A2 right at the beginning when arachidonic acid was being made. Then they can also block the elaboration of various cytokines. That is why if you have been a little aware, dexamethasone was actually touted as one of the frontline treatments for COVID-19 because it is a broad spectrum inhibitor of the inflammatory response. There are also other ways in which you can inhibit the eicosanoid synthesis. So you modify the intake and content of dietary lipids, and then you increase the consumption of fish oil. So you increase PUFA consumption. This is a poor substrate for conversion to downstream active metabolites. And there are other molecules called resolvins and protectins, which also are excellent molecules for totally suppressing the inflammatory response. Now, this is a chart by which you can see what is the action and what is the molecule. So if you look at vasoconstriction, which I earlier talked about, vasoconstriction, two groups, TXA2 and LTCDE. The rest are all vasodilatory. That is, the prostaglandin family is largely vasodilatory. Then you have increased vascular permeability, that is LTC4D4E4, and you have chemotaxis, that is LTB4. Okay. So there's another thing now in that long list, and that is called as platelet activating factor. So it's a phospholipid derived mediator. It has multiple inflammatory effects, and basically ABC is just referring to it causes aggregation, it causes bronchoconstriction, and it causes vasoconstriction. Okay, so it's another one of the vasoconstrictive molecules. A platelet activating factor at extremely low concentrations can go against its basic nature. So there it's going to become a vasodilatory molecule. Otherwise, it can also cause adhesion, chemotaxis, degranulation, and the oxidative burst. This is important. You have reactive oxygen species. Now, reactive oxygen species, or ROS, any inflammatory response in the body, and you have the generation of the ROS. So what is ROS? Basically, these are oxygen-derived free radicals, and they are extracellularly elaborated from the leukocytes. You give any trigger to the leukocyte, especially the neutrophils, and then they start elaborating these oxygen-derived free radicals. The production is definitely dependent upon the NADPH oxidase system. And what do you get? You get all these ions that are produced: superoxide, H2O2, hydroxyl radical, and these are the molecules that are produced within cells, and this is capable of causing widespread inflammation. So it is going to wreak havoc. It is going to damage your endothelial cells that are lining the blood vessels. So when you damage the lining of the blood vessels of the body, then there is increased vascular permeability because the cell lining gets destroyed. There are gaps in the cell's lining. So if a fence is there, if you can imagine a fence that is lining a blood vessel, and if it's destroyed, then there are going to be holes. So this is going to result in increased vascular permeability, right? Then second is injury to other cell types. So it's going to cause injury to other cells also. Apart from the vascular compartment, other cell types in the body, parenchymal cells, other cells are also going to be affected by ROS. And it is going to inactivate antiproteases. So it's going to inactivate the good guys. Now, antioxidants, apart from, so this is ROS. Now, can we do anything to stop ROS? Yes, we can. We can use antioxidants. And that is why you can see many people use a lot of supplements in the diet to kind of, delay aging or delay other, not just for health and wellness, but even in disease processes. You have supplementations given by physicians that can halt ROS, so you halt the overall inflammatory response. So you have SOD, you have catalase, glutathione peroxidase is commonly prescribed supplement. Okay, then ceruloplasmin and serum transference. So all of these basically are antioxidants. Now NO. Now in context of inflammation, I would say NO is what it is. NO means no. Okay. What is no? No means no inflammation. So it was discovered as a factor released from endothelial cells. Now, if you notice very carefully, in any step of acute inflammation, everyone is talking about the vessels. The vascular component is very, very important. So it's discovered as a factor released from endothelial cells, causes vasodilation. Remember, most of these molecules are causing vasodilation except for the vasoconstrictive thromboxane and LTCDE. Okay. Now, and of course, we just read platelet activating factor. Now, you have NO is a soluble gas which is produced by endothelial cells, and it acts in a paracrine manner on target cells. So it's like a local friend of yours. I act and I'm going to make the person beside me act in a similar way. Causes relaxation of the vascular smooth muscles. Now, NO has dual actions in inflammation in the sense that it promotes vasodilation, it relaxes the vascular smooth muscle, doesn't cause contraction, and it is an inhibitor of the cellular component of inflammatory responses. Okay, so as I said, NO for me means no, so it's reducing platelet aggregation and adhesion. It is inhibiting several factors, several features of the overall inflammatory response. That is your antigen antibody mediated reactions, which are typically called as the mast cell induced inflammation. NO is also inhibiting leukocyte recruitment, so the soldiers aren't recruited either. And NO is doing one other good service, it is killing the microbes. So NO is a mediator of host defense against infection. Now, we come to something that is extremely important, and that is the cytokines and the chemokines. Cytokines are produced by many cell types. In fact, I would just go one step further and tell you that cytokines can be produced by every cell type. So they are principally activated lymphocyte, elaborated by activated lymphocytes and macrophages, as well as all the extracellular compartment cells like connective tissue cells and cellular like epithelial and endothelial cells. So nowadays, every exam, even undergraduates, can be asked this thing about the cytokine storm of COVID-19. So you have to really read this with an open mind. So cytokines and chemokines, tumor necrosis factor and IL-1. These are the major, I would say, the major couple involved in inflammation. They're produced by activated macrophages, and their secretion can be stimulated by all types of injurious stimulus: physical, heat-induced, traumatic, microbes, you name it, you got it, and the cytokines are elaborated. Okay, then this is just a sort of a synoptic review of the principal local and systemic actions of this couple, TNF and IL-1. So what is this couple doing? Macrophages are activated. Now, when are macrophages activated? When you get a dozen a dime injurious stimuli in the body. After that, macrophage activation occurs. After macrophage activation occurs, the couple arrives, TNF and IL-1. What does the couple do? It causes either local effects or systemic effects. Systemic effects of inflammation are only of one hue, that is fever. Then you would have an increased WBC count, you would have acute phase protein elaboration, the sleep would go, over here they've said sleep is increasing. I'll just have to check on that. From my mind, the patients should actually, they should not be sleeping, but yeah, I'll just check on this. So sleep and appetite is definitely decreased. Okay, so then this is part of the septic response, and an elaboration of TNF and IL-1 is going to cause cachexia also. So there is weight loss. This is part of the septic response, and this is called as systemic manifestation of inflammation. TNF IL-1 couple can also cause local effects. So the local effects would be the WBCs are going to be, incited. Come on, produce more cytokines. The fibroblasts. What are fibroblasts? Fibroblasts are mesenchymal cells, cells outside the actual cells. So the extracellular compartment. Now, what are fibroblasts going to do? They're also going to get activated. And when fibroblasts are going to get activated, they produce collagen. And when collagen is laid down in the body, it promotes healing and repair. So this is part of the repair response. Another effect is, as I said, no talk about inflammation can be successful without talking about the vessel. So the vessel is lined by endothelium. Now, when the vascular endothelium is activated, what happens? They, in turn, it's a vicious cycle. They produce more TNF-IL-1. They produce more procoagulants, and they produce leukocyte adhesion molecules, which in turn promotes more inflammation. So this is talking about the couple. So in the endothelium, when the endothelium is activated, you have endothelial adhesion molecule activation, and you have synthesis of the various chemical mediators, the entire list I've talked about. Second thing is that you would have endothelial activation results in production of enzymes associated with matrix remodeling. What does this mean? Nothing. It means one thing, that the moment you have an activation of an endothelial cell, you are also going to activate the immediate neighborhood. As I said, the immediate neighborhood is nothing but it is the matrix, which is the extracellular matrix. And the extracellular matrix is then going to be activated, and it is going to remodel itself in order to give a befitting host response. The third thing is that the surface thrombogenicity of the endothelium increases. And as I said, everything is a vicious cycle. So this also, it further activates. The neutrophils are totally in that zone now. They're totally geared for the battle, and their response to other stimuli is also heightened. So, as I already mentioned, they induce the systemic acute phase responses also. Right now, they suppress the appetite. That's because the energy balance is totally gone. You promote lipid and protein mobilization, the appetite is suppressed, and with all this activity happening, so much of energy expenditure is going on in the body naturally with the elaboration of so many molecules. So the net result is very obvious. You're going to lose weight, and that is called as cachexia. So this is basically summarizing what I just talked about now. Activation of macrophages, elaboration of the couple. The couple is leading to the systemic response, the effects at the level of the endothelium, the fibroblast effect, and the leukocyte effects. So chemokines. Now chemokines are actually a family of smaller proteins, but don't underestimate their capacity. They have two functions. They are also going to recruit leukocytes, and they organize the cell types in various tissues. Now my experience with chemokines is, very frankly, as a student, in my student life, I only saw them in some sort of competitive entrance test, but they're always in front of your eyes in some form of MCQ or the other. So take it with a pinch of salt, but also listen to what's going on. So they classify into four major groups. How are these chemokines grouped? They are grouped according to the arrangement of the conserved cysteine residues. So now just look at the figure. CXC chemokine. X is the orange thing. Now that is an amino acid residue, and it is separating the pair of conserved cysteine residues. So CXC chemokine, example is IL-8, acting primarily on the neutrophils. And it's going to help in neutrophil activation, chemotaxis, and the works. There is a CXC receptor 4, and that is binding HIV. Basically, that is the MCQ that is generally asked. Then the other MCQ that is asked is, IL-8 is what kind of a chemokine? So CXC, okay. Then you have the second group, that is CC chemokines. So again, look at the figure. You have two conserved cysteine residues that are adjoining each other, pairs, right? And the examples of this are MCP1, eotaxin, rantis, and MIP1 alpha. Now I put this as merm. So you have a mermaid over here, and MCP1, eotaxin, rantis, and MIP1 alpha. Okay, so these are the examples of the CC chemokines, which have two pairs each of conserved cysteine residues. Yeah, and just a sec. Going back, this is another MCQ. So CC receptor 5, again HIV. So that is another MC. Third one is the gamma chemokine or the C chemokine. So there is no, they lack the first and the third of the four conserved cysteines. Example is lymphotactin, and as the name suggests, it's specific for lymphocytes. Then you have CX3C chemokines. So if you look at the figure, again, you have three amino acids between two cysteines, and the example of this is fractalkine, right? So this is all that you need to know about the chemokines. Let's go into another class. So this is the lysosomal constituents of leukocytes. Neutrophils and monocytes have lysosomal granules. We all know that. Basically, all cells have lysosomes, so they are no different. And neutrophils have two main types of granules. So one is the primary granule, and one is the secondary granule. So primary granules are always large, because primary means first, so primary large granules, and smaller secondary and specific. If you see SSS, that's how I remember it. So smaller granules, specific granules, secondary granules. Larger granules are primary granules. Now neutrophils, in hematology when you study blood and you see these blood cells only. So sometimes you have to pick up neutrophils in special situations, and neutrophils make a very important enzyme called the myeloperoxidase. So this is elaborated by the primary granules. So if you can see on the right, you have a picture that is nothing but an electron microscopic image or an ultrastructural image. So you have normal microscopy in which I showed H&E stained sections last time. Sometimes you need to increase the resolution and go deeper and deeper within the cell, and you need a very high resolution to see the endoplasmic reticulum, to see the cellular organelles. That objective is achieved by the electron microscope or the ultrastructural analysis. So you can see over here that you have nuclei, and these darker blobs over here are the primary granules within the neutrophils. Okay. Now again, so as I said, you can have the two types of granules. They can fuse with phagocytic vacuoles. They can be released into the extracellular space. So whatever the neutrophil is elaborating may remain within the cell itself or may be chucked out of the cell and released into the extracellular space. What is this doing? What objective are we achieving? Well, we're going to activate the complement system, which is a very important category of mediators of inflammation. Secondly, naturally, a mediator of inflammation is going to produce tissue injury, and obviously it also would enhance vascular permeability. So this is a short one, neuropeptides. So I've underlined the word peptide to just pep. So this is what its action is. It's pain signals. It has an endocrine activity. It regulates the blood pressure and it increases vascular permeability. Some things you just have to remember verbatim. And it is neuro, so it is secreted by nerves. It is secreted by nerves which are of the GIT, of the RESP, and by various leukocytes. And neuropeptides, remember PEP, and you would remember the biological functions. So now coming to the last category of chemical mediators of inflammation, and that is the plasma proteases. So as I said, cafe, coffee day, cafe, coffee K. And you have a variety of phenomena in the inflammatory response that are going to be mediated by these people only. So we've talked about the cell-derived. Whatever was being talked about till now was all the cell-derived mediators. Whatever we shall talk about now are going to be the plasma protein-derived mediators. It is the liver that is the major source of these molecules. And basically, you would have the elaboration of factor 12 or Hagemann factor, which is going to result in the production of the kinins and factors in the coagulation system, as well as complement activation. So C3A, 5A, C5B, these are all important molecules of the complement system. Okay. Yeah. Divyanshi, can you mute yourself? Yeah. Okay, so now you have plasma protein derived mediators. So as I said, complement, clotting and kinin systems. The complement system, we're numbering it through C1 to C9. And again, the same business. It is going to increase the permeability of our vessels. It is going to help in recruitment activities or chemotaxis. And it is going to make the bacteria more tasty to eat. So that is called opsonization. And a critical step in complement activation is the proteolysis of C3, that is the most abundant component. So functionally, you have three general categories. You have C3A, you have C5A, and you have C4A. What are these people doing? They are helping in stimulating histamine release from the mast cells. So as I mentioned earlier, either histamine release is going to be because of some injurious stimulus, some antigen antibody binding, and thirdly also by the complement system. So that is mediated by these molecules. C5A is a very powerful chemotactic agent. That is also asked as an MCQ. So that is which agent is a very powerful chemotactic agent? The answer is C5a. And C5a also helps in the LOX pathway. So if C5a is being activated, then the LOX is being activated, and LOX results in all your leukotrienes to be formed. So again, more inflammatory mediators. Another thing, so they wreak havoc by causing the elaboration of more mediators. Secondly, they wreak havoc by activating the phagocytic system. So that's in a way a good thing. So you promote phagocytosis by neutrophils and macrophages. So you're trying to contain the offending agent. Cell lysis. Now ultimately the cell which has all this going on has to die. So deposition of membrane attack complex on the cells, which results in the death or the lysis of the cell. So as I mentioned, the storyline is all about C3a, C5a. These are the most important molecules. They can be cleaved by several enzymes in the inflammatory exudate, and this results in a vicious cycle of neutrophil recruitment. So in Robbins, this is an important diagram, and that is the activation and functions of the complement system. So activation of complement by different pathways. See, everyone is interested in cleaving C3. That is the end result of everything. So you have a classic pathway, you have an antibody binding onto an antigen, you can see, and C3b is deposited on the microbe, making it very tasty for phagocytosis, right? Then you have alternate pathways like lectin binding. So mannose binding lectin would also come onto the surface. Net result is the cleavage of C3. Once C3 has been cleaved, you get C3a, C3b, okay? And C3a, as we've talked about, is helpful in recruitment activities and further destruction of the microbes. And C3b helps in formation of the membrane attack complex leading to lysis, and C3b helps in phagocytosis by recognition of bound C3b by the phagocyte C3b receptor. Now coagulation. So this is the last point, coagulation and the kinin systems. Culmination in basically the end point over here is that we are making thrombin, and therefore we are making fibrin, and we are blocking the sort of blood or bleeding response that is associated with inflammation, and this is constituted by a series of plasma proteins which at the outset I said were activated by Hagemann factor or factor 12, which is an important factor elaborated by the liver. So you have, this is factor 12. We start over here, factor 12. Now factor 12, when it is elaborated, comes in contact with the activated platelets, collagen, basement membrane. I showed you one diagram of the activated platelet in serotonin. So similarly, factor 12 gets in contact with that environment and it gets activated. Factor 12 becomes factor 12A. Okay, and in its activated form, it is going to help in doing two things. It is going to help in activating the clotting cascade. So that is nothing but activation and elaboration of thrombin. What is thrombin doing? It is converting fibrinogen to fibrin, and fibrin split products are formed. Secondly, you have factor 12A that is helping in the conversion of free kallikrein to kallikrein, and kallikrein results in the production of kinins. I know it's slightly difficult to understand at this level, but for the moment, all you can understand is factor 12 is the Hagemann factor. When it gets in contact with collagen, the basement membrane, and activated platelets, it gets activated. Then, of course, it results in the clotting cascade. How? By the production of thrombin. Also results in the conversion of free kallikrein to kallikrein, and this results in downstream kinin production. So, what are kinins? They're nothing but vasoactive peptides, and the important one is bradykinin. So that is the very important one. Bradykinin increases your vascular permeability. Bradykinin causes smooth muscle contraction and dilation of the blood vessels, and it results in pain when injected into skin. Bradykinin is typically associated with the pain response, and it is very, very short-lived. Now, apart from that, factor 12A, I mentioned earlier, is inducing the fibrin clot formation, and basically it helps in, it is going to cause the activation of the fibrinolytic system. Okay, so kallikrein, as I mentioned earlier, this is actually leading to downstream kinin production by cleaving plasminogen. So now in the fibrinolytic system, what is the primary function of plasmin? It is the lysis of fibrin clots, cleaving complement C3 to produce your C3 fibrins, and of course, then it results again in the activation of the Hageman factor. So to summarize all this, bradykinin, C3A and C5A, these are all causing increased vascular permeability. As I mentioned, bradykinin also has an important role in the pain response. C5A is a mediator, one of the most potent mediators of chemotaxis. Thrombin is basically going to help in formation of your fibrin clot, and it has an effect on the endothelial cells. So C3A, C5A, most important complement components, and it results in immunological reactions. It also results in activation of the alternative and lectin complement pathways. So if we recapitulate in that diagram, we had three things going on. One was the classical pathway, one was the alternative pathway, and the lectin complement pathway. Okay, so basically then there are agents that are not directly related to all this mumbo jumbo, and that is the plasmin, the kallikrein family, and some of the serine proteases. Okay, so I think, so this is just a summary of, you have factor 12A, it has a relationship with the kinin system, the clotting system, the fibrinolytic system. Fibrinolytic system is producing plasmin, which is degrading the fibrin, okay, and then you have the complement system. So again, this is just summarizing what all we've discussed. I won't repeat myself, but you can maybe take a screenshot, and I'm going to upload the presentation in any case, so you can study these pathways and recapitulate them again and again and again, because that is what is required actually. Okay, so yes, so you have all this going on. We've talked about it. We have also talked about the various pathways by which the complement system is going to be activated. Our focus is primarily on C3, that is the key complement thing. Just to summarize again, C3, C5A, most important, 5A chemotaxis, 3B opsonization, and 5 to 9 involved in the membrane attack complex formation. Is there a role of mediators in different reactions of inflammation? Yes. So if we talk about vasodilation, already mentioned that. I should have added vasoconstriction in this slide also, but I've already talked about that earlier. Increased vascular permeability is definitely by most of them. Chemotaxis, C5A is the important one, chemokines and LTB4. Fever is by the couple, TNF, IL-1 as well as PGs. Pain is by bradykinin and prostaglandins, and tissue damage is basically by ROS and the neutrophil and macrophage lysosomal enzymes. Now we have one gross over here, and Anshika, can you tell me what this is? The last slide today. Can you tell me what this is? I want a little interaction now. I've been speaking non-stop. So Anshika, can you tell me what this is? You don't know, or you're not here, or are you here? This is interesting. Okay, Asna. I can see Asna. Yes, Peter. So what is this? Any guesses? Huh? What do you think this is? Yeah? What do you think this is?
A kidney.
Fantastic. How did you
I
find
guess
out
this
that
was
it was the kidney?
kidney.
Okay, so how did you find out this was the kidney?
Ma'am, because of its membranal pyramids.
Okay, very good. You're a very intelligent group of students. So basically, when you describe any gross specimen, you have to say that it's a formalin fixed wet mount. Okay, formalin fixed